My name is Adrija Mamidipalli. I'm from University of California, San Diego, Liver Imaging Group. I'm going to talk about a recent publication in the Journal of Magnetic Resonance Imaging, cross-sectional relationship between hepatic proton density fat fraction and hepatic R2 star estimated using MRI-C and MRI-M in children with non-alcoholic fatty liver disease. I'll be only presenting brief highlights of the paper. For detailed information, please read the paper. These are our disclosures. Quantitative imaging biomarkers for characterizing non-alcoholic fatty liver disease are becoming available, including in children. Two such biomarkers are proton density fat fraction (PDFF) and R2 star. PDFF is a non-invasive biomarker of fat, and R2 star is a non-invasive biomarker of iron. Both these biomarkers can be estimated simultaneously by chemical shift encoded, multiecho, gradient echo MRI sequences. The underlying hypothesis is that iron particles are small magnetic dipoles that alter the local magnetic field, accelerating T2 star decay. Other objects that can act as small magnetic dipoles may also accelerate T2 star decay, and one potential example is intrahepatic lipid droplets. Therefore, we hypothesize that fat droplets may accelerate T2 star decay, that is, fat may increase R2 star, which is 1 over T2 star. If so, then PDFF and R2 star will be correlated in human liver. This is a normal liver which contains water and magnetic field is fairly homogeneous, and this is its slope of signal decay. Then we put iron particles which have strong dipolar field that causes magnetic heterogeneity, and there is more rapid decay as illustrated here. And then fatty liver with fat droplets that have a weak dipolar field, less magnetic heterogeneity compared to iron, and a rapid signal decay as shown here as compared to that of normal liver. Therefore, the purpose of this study is to explore our hypothesis that hepatic R2 star and hepatic PDFF are correlated in human liver. And for this study, we focused on children with known or suspected NAFLD, as they have a wide range of steatosis severity, they rarely have hepatic iron deposition, and hence are appropriate experimental model to explore our hypothesis. Coming to methods, this is a retrospective cross-sectional analysis of research MR exams acquired prospectively at UCSD with GE scanner in children with known or suspected NAFLD. Two MRI techniques were performed, and R2 star and PDFF were estimated simultaneously with these two techniques. The reason we used both techniques was to make sure that our results were not technique dependent. Complex-based MRI and magnitude-based MRI are the two main techniques used in this study. Complex MRI uses both magnitude and phase information and can estimate fat fraction maps from 0 to 100% and R2 star values, while magnitude MRI uses only magnitude data and can estimate fat fraction maps from 0 to 50% and R2 star values. For complex and magnitude MRI R2 star and PDFF analysis, we deposited 1 cm radius regions of interest on 5th echo magnitude images on all 9 Couinaud liver segments, which were then propagated onto reconstructed PDFF and R2 star maps as shown here. PDFF and R2 star from each ROI was recorded from the map, from which mean PDFF and R2 star values were calculated. And then Pearson correlation coefficients were computed between R2 star and PDFF for both MRI-C and MRI-M techniques. And the two correlations, R2 star versus PDFF with MRI-C and R2 star with PDFF with MRI-M, were compared using Steiger's test. And finally, Bland-Altman plots comparing MRI-C and MRI-M for R2 star and PDFF estimation were computed. We enrolled a total of 184 children, boys more than girls, as seen here, which is typical for pediatric NAFLD. Notice that there is a wide range of PDFF with both MRI techniques and both for boys and girls. And notice here that the range of R2 star was below ion overload values with both MRI techniques at 3T, suggesting that these kids have no ion overload. This is a linear regression plot between hepatic proton density fat fraction on x-axis and R2 star on y-axis estimated by complex-based MRI in red and magnitude-based MRI in blue. This plot is showing a strong and significant correlation between hepatic R2 star and hepatic PDFF by both MRI techniques, and the Steiger's p-value for the two correlations was not different as shown here. This figure is showing hepatic R2 star and PDFF maps of 3 representative pediatric participants with low PDFF and low R2 star values, medium PDFF and medium R2 star values, high PDFF and even higher R2 star values, illustrating the relationship between R2 star and PDFF with both MRI-M and MRI-C techniques. This Bland-Altman plot is illustrating the difference between hepatic R2 star estimated by MRI-M and MRI-C as a function of average R2 star. This plot demonstrates a close agreement between the two MRI techniques across a wide range of R2 star with a small but significant bias of 6.16 second inverse MRI-M overestimating compared to MRI-C. Standard deviation of MRI-M and MRI-C differences and limits of agreement are overlaid in the plot. And this Bland-Altman plot is illustrating the difference between hepatic PDFF estimated by MRI-M and MRI-C as a function of average PDFF. This plot demonstrates a close agreement between the two MRI techniques across a wide range of PDFF with a very small but significant bias of -0.62% MRI-M underestimating compared to MRI-C. Standard deviation and limits of agreement are overlaid in the plot. Our results confirmed our hypothesis that R2 star and PDFF are correlated in human liver independent of imaging technique. But the mechanism behind this correlation is not proven in this study. One most likely explanation could be fat droplets acting as magnetic dipoles inducing local susceptibility effects accelerating R2 star decay of neighboring water molecules. Other possible mechanisms that are unlikely are different R2 star for fat and water, which is unlikely to be true as a recent study has shown that R2 star of fat and water are similar in the current fat spectral model used for PDFF analysis with MRI techniques. Confounding by ion overload, which is also unlikely as children with NAFLD rarely have ion overload, and the observed ranges of R2 star at 3T in our study are not suggestive of meaningful ion overload. Our study cannot exclude the possibility that the relationship is mediated by histological factors other than ion, as histology was not available. Therefore, further research is needed to examine the postulated role of fat droplet-induced susceptibility as well as explore other possible mechanisms. If confirmed by future studies, our results suggest that PDFF value should be considered when interpreting R2 star as a non-invasive biomarker of hepatic ion overload, and PDFF correction of R2 star measurements may be needed to reduce the confounding effects. Caution is suggested in applying MRI-C and MRI-M interchangeably for R2 star and PDFF estimation, as there were small but statistically significant differences in their estimation by the two techniques. The following are our study's limitations. This was a single-center, single-scanner study including only children without histology data. In conclusion, R2 star and PDFF are strongly correlated to each other in a patient population that is known to have rare hepatic iron deposition. The relationship was observed for two techniques, MRI-C and MRI-M, suggesting that this is not a technique-dependent finding. And further research is needed to verify our findings in independent cohorts, including adults, as well as using different types of scanners and field strengths. Thank you.